think we're live. Okay, so good morning. This is my first live lesson. Excited. Bear with me while I get used to this. If you have questions, please ask. I have it set up here that I can see you've asked questions and I'll address them. So, as is the first live one, I wanted to do something that is not too much because, again, it's the first for me live lesson, so bear with me. Okay, so I'm going to do experiments first. The questions that would be with question 8. So to remind you, section B is worth 15% of your actual leaving cert. There's 3 questions, answer 2, and those 2 go towards your marks. If you want to answer 3, go ahead and answer 3. Your best 2 are selected. So I'm going to go through the questions, each experiment. And again, if you have any questions, please ask. Even if you think it's silly, if it's not, no such thing. Okay, so the first question is with food. The first experiments we're going to look at is with food. So they can ask you to test for the presence of starch. What you do need to know is you need to have iodine. Okay, so that's your solution, your chemical reagent. So I have here, add a few drops of your iodine solution to, it could be a food source, so a bread, a piece of cracker. Or some of you actually may have a starch solution either. But what we're looking for is that we have iodine. Your result, if starch is present, it'll go blue-black. If it's your control is always usually going to be water. Your control will stay the same color as your iodine, will be yellow-brown. So I just have here the same pictures that are in our textbook, okay, so from book 1, and this is just a little table here. So starch, what we're going to use, it's a starch solution. We're going to use iodine is what we're going to test for starch. If starch is present on our sample, it's going to go blue-black. Our control is water, so the iodine will just stay the same. No questions just yet, okay. To test for a fat, okay, so you're going to have brown paper for this, and you're either going to rub oil, you might use a bit of oil, or you might use butter. So what you're going to do, rub it onto a piece of your food sample, your fat, whether it's oil or butter, onto your brown paper. Your control will be water, and then you leave them to the side to dry. And if fat is present, it appears translucent. What will happen, you're negative with your water, it'll dry out and it'll just return back to what it was. So you usually get your brown piece of paper and you'll hold it up towards the light. So that's your test for fat. And this is just, again, the same picture, that we have in our textbook. Our reducing sugar. So with your reducing sugar, the reagent that you use, the chemical that you use is Benedict's. Now I'm saying the word reagent because they might phrase it, what is the reagent you use to test for the presence of a reducing sugar? And you put your Benedict's, it's blue in color, and you would add some of that to your test tube where you would have maybe a glucose solution, and then you would heat it. So this is the only food test that requires heat. Now, I would say heat at 60 degrees. Do not say heat on its own, because what's hot to you may not be hot to me. So let them know we want something that's above 60 degrees. So you're going to heat it in the water bath for about 10 minutes, 10 to 15 minutes. If it's positive for reducing sugar, it'll go from blue to brick red. So remember, blue is the reagent. Blue is Benedict's at first. So that's, if your teacher had given you the Benedict's, it'll be blue in color. I keep looking this way because that's where my phone is for the questions. But I have no questions just yet. Okay, your control. So your control will be water in a test tube, and to that we add our Benedict's. You would also put that into your water bath. Again, your water bath, you're going to heat it at 60 degrees for 10 minutes. I would also include the time. Okay, so if asked that, it's heat at 60 degrees and the time frame. Because if you said you left it there for a couple of minutes, what's a couple of minutes? You're not going to get a positive test after a couple of minutes. You're just not. And actually it can be about maybe 15 minutes. So when you first would do this, it would go orange first, then green, and then eventually it will go brick red. So your control will stay blue. So the test for reducing sugar is the only one that requires heat. Okay. Your test for protein. So your chemical that we use, the reagent, is burette. It's also blue in colour. Now, you can say here it's sodium hydroxide and copper sulfate. But what is easier for you to learn? Is it easy for you to learn to test for protein is burette, or is it easy for you to learn sodium hydroxide and copper sulfate? You can use the chemical formula for that if you want either. So a positive test, so I would get milk, that's what I would use in school, put milk into the test tube, that's my source of my protein. I'd add my burette, which is blue in colour, shake that, and it will go, if it's positive for protein, it'll go purple. Now, you'll also accept here if you say violet, if you say lilac, if you say mauve, but purple is probably the easier one. So we'll go from blue to purple if it's positive for the protein. Your control will be water, add your reagent blue, give it a shake, and it will still stay blue. Okay, so that's your food tests. Now, food tests can also get asked with question 1 or question 2 with food. So if, for example, you're getting question on protein, they could also put this in here. So don't ignore the food tests at all. Sorry, just keep looking there. No questions. Okay, so now this brings us on to ecology. So while ecology is also in question 8, ecology could be questioned with this in section A, and it could be questioned in section C. It could be A, B, and C. So ecology is important. So the first thing you have to identify is 5 animals and 5 plants, so your flora and your fauna. What do you use to identify it? You use a key. And I've just reminded you what a key is, so answering a series of questions. And then your flora is just your plants, your fauna is your animals. The ecosystem I would do in school is a woodland, so these are just some flora and fauna that are familiar in a woodland or grassland habitat. So what you need to know now, if they ask you what do you use to identify flora or fauna, you can say a key. I put here as well, they'll also actually accept an app. Okay, identification app. So if you just said an identification app, this is a great one actually as well. Here, see, I believe it's free that you can download. You can take a picture of your plants and your animals, and it will come up with the name of it. So they will accept a key and they will accept an identification app. So you may be asked to draw a key for animals and for plants. So if you have followed me and we did the ecology and I uploaded the field study questions, I think this is of the 10 questions that I put together. I think this is question 1. It says name 5 animals. It's either question 1 or question 2. Name 5 animals found in your ecosystem, draw a key to identify them. Name 5 plants found in your ecosystem, draw a key to identify them. So this will be a very simple one here. So here's my animals. So does the animal have a backbone? No, go to 2. The animal does it have a backbone? Go to 2 if it does. If it doesn't, go to 3. So animal has feathers, therefore it's going to be the blackbird. Has no feathers, it's going to be a frog. Animal has legs, so it's our bumblebee. Has no legs, go to 4. It has a shell, so therefore it's a snail. It has an earthworm, has no shell, earthworm. Then we have for plants as well. So 5 plants: buttercup, daisy, thistle, dandelion, and nettle here. Now, I think, in my opinion, it wouldn't be nice to get asked this question. Have they done it before? Yes. Just keep it simple if they do. So your plant. So a flower is a yellow, at least in some parts, go to 2. A flower is not yellow or it's absent of colour, go to 4. So flower with white petals is your daisy. Flower with yellow petals, go to 3. On 3, excuse me, flower with 5 petals is your buttercup. Flower with more than 5 petals, your dandelion. Number 4, flower is pink and purple, it's your thistle. Flower is absent or green, it's your nettle. Okay, so you have to be able to identify a variety of habitats within your ecosystem. So we need to know 3. So reminding you, what is a habitat? It's a place where organisms live. Possible habitats that I would do within my ecosystem is ground view, so what you see. Litter habitat, so your leaf litter, move the leaves. And then we have our soil habitat. Excuse me. So that for this, just what is your habitat and 3 possible habitats they may ask you. Then we need to be able to know 5 pieces of apparatus to collect your organisms. Now, reminding you, it's collection. Direct search is not collecting, it's what you see. Okay, so what are you actually putting these organisms into? So you have your pooter, for example, your beating tray, pitfall trap, sweep net, and the Tullgrin funnel. I did mention before, with the Tullgrin funnel 2 years ago, was asked students, not many at all actually knew what a Tullgrin funnel was. So your pooter, how does it work? So you suck through the mesh tube, and what we'll do is collect organisms with inside. Your beating tray, you have a white sheet, you might just have a normal stick, and you hit your tree or your bush, or whatever it is, 3 times. I would say 1 on the first hit, so the insects like that, they're caught off guard, they'll try to cling on. 2, the second hit, and the insects again will try to cling on. The 3, they're going to fall off. So it's white because we want to be able to see what we're actually have caught. So insects, caterpillars, spiders, lots of different things there. Your pitfall trap. If you're asked to draw this, make sure that your pitfall trap is in line, your jar, whatever it is in line with the soil, so that whatever it is walking along can fall in. So make sure that there's no gaps. Okay, so that would be important. So we would put a stone or something above it, a piece of wood, whatever you find within your area, within your habitat. So if it rains, that if something was caught inside, that we're not going to kill it and drown it. Your sweeping net, ideally that would be through long grass. Would you draw a sweeping net? And again, you might catch stuff in the sweeping net, and then put the contents of that onto a white sheet. Again, a white sheet for us to be able to identify. If you didn't know how to identify it, use a key, or you can use that app. And I have a question. Kevin, do you need to be able to, just bear with me, sorry. Okay, good question. So Kevin's asked, do we need to be able to label it if we're asked to draw a pitfall trap? If you're asked to draw any diagram, you label it, okay? If it says sketch a pitfall trap, you don't need to label it. Sketch anaphase, you don't need to label it. So if you're asked to draw, you label, always label. And if it said label 3 things, I would label 5, okay? But draw a diagram, you label. Sketch a diagram, you don't have to label. Good question. Thanks, Kevin. You're welcome. Okay, very good question actually, regarding any diagram. Label. And you don't have to be an artist. I'm not an artist. You don't have to be an artist. You don't have to color in a diagram. You don't have to shade it in a diagram. So when we're labeling diagrams, we're looking at first the diagram. Does it look as what it's meant to be? And then we'll look at the labels. Are they labeled in the correct areas of what they're meant to be labeled? So then your Tullgrin funnel. Now, they might be sneaky and throw this back in again to see, have people looked at the past papers? Have people seen that this was badly answered? So what I would do for this, the Tullgrin funnel in class, is we don't do it. I would get a sample of soil from our habitat, from our ecosystem, and I would bring it back into the class, and I'd set this up with the students. So the Tullgrin funnel has a light source, and what the insects will do is bury themselves away from the light source. So you'll have it literally looks like a gauze at the end here with little holes in it, and the insects will fall down into that. So Tullgrin funnel for that. I don't know, maybe they will or they won't, but just to be aware. Okay, they love asking this. To carry out a quantitative survey of plants, and this one, for example, is percentage frequency. Quantitative records the number of organisms present. Qualitative, whether the organisms are present or absent. You need to know those 2 definitions. So with your quadrat, it doesn't matter if it's a gridded or quadrat or just a normal metal quadrat, excuse me. I should have brought a glass of water. So what are we going to do? So there can be at least, I would tell my students, I want at least 7 points for this. So the first thing you're going to do is you're going to throw a pen over your shoulder. Why? It's random, and say it's random. for safety, and it reduces bias. So there will be a mark for that throwing the pen over the shoulder. Then we're going to place your quadrat down where your pen has landed. And for saying quadrat, there's a mark there for a quadrat. You're going to record whether your named plant is present or absent. You're going to repeat this for 9 more throws, so you've done it 10 times. Then you're going to calculate out your percentage frequency, present your results in a table, and then you're going to plot those. With this question, it might say, carry out a quantitative survey of named plants in your habitat. So there could be a sneaky mark there going for the named plant. Grass, daisy, keep it really, really simple. So throw a pen over your shoulder. Why? It's random, reduces bias, safety. Place your quadrat, a mark for your quadrat down. Record whether your plant is present or absent. There's a mark there going for record. Repeat, there's a mark, repeat. Repeat this 9 more times. Calculate out the percentage frequency. Present your results in a table and then plot results on a graph. This is what you need if this question is asked. So then here's our table of our results. So we're calculating out your frequency. They're not going to ask you this, I've just put it in here. So it's a chance of finding a named species in one throw of a quadrat. There are disadvantages they might ask, I don't know. So it's dependent on the organism and the quadrat size, and assume that organisms are evenly distributed. That's what we're assuming. So you record it 10 times here, you calculate out your percentage frequency. This table here, would they ask you? No, but they might show you, or maybe in a mock, a table like this and ask you to calculate out the percentage frequency. So it's your number of quadrats with your organism by the number of throws. So number of quadrat throws. So here's a table showing our results. Conduct a quantitative survey of plants in an ecosystem, your percentage cover. Now, percentage cover is an estimate of the ground in which the quadrat covers each species. To be really honest, the percentage cover and percentage frequency, within writing it up, there's no difference in writing it up, other than with the gridded with the percentage cover, you're going to touch your pencil down at the gridded points. If they ask, usually it is conduct a quantitative survey of plants. They aren't as specific now regarding percentage frequency or percentage cover. So the same method, throw your pen over your shoulder. Why? It's random, reduces bias, and safety. There's marks there for that, at least 3 marks there. Place your quadrat down, a mark for your quadrat. Using your pencil, I have them here, push at the gridded points and record if the plant is present or not. So when I do this in school, I would get my students to go, if it's my gridded quadrat here, I'd get my students to go to the right-hand corner and see if it's present or absent. So then again, the same thing. We're going to repeat this 10 more times, calculate out the percentage cover. You're going to present your results in a table and then plot your results in a graph. So marks are going for, and the same with percentage frequency. Your marks are going for mentioning graph, for presenting results in a table, for possibly saying percentage cover, definitely for repeating, definitely for recording, throwing over your shoulder, definitely for saying it's random, bias, or the marking scheme might say to us, how is this random? Placing mark, we're going for quadrat, and then you're going to record if the plant is present or absent. And the same thing again, conduct a quantitative survey of plants looking at, or could say named plants. And again, just keep it really, really simple. So here I have my results. So there's 25 points in a gridded quadrat. So that's why we have here multiplied by 250 times by 10. Sorry. So that's your results there. I don't see them giving you this and asking you to calculate it out. This is where the marks are going for. So number of hits by 100. So total number of points. That total number of points is your 250. To carry out a quantitative survey of animals. So just read, if that question is to appear, just read it slowly. Are they asking you for fauna or flora? Okay, are they asking for animals or for plants? If it's animals, there's marks going for saying this is your capture, recapture. There's marks there for saying capture, recapture. And we'll ask you for a named animal. And the easier one is to say snail. Don't say anything that's going to fly away. So on your first visit, so you must make reference to this. Now, the answering of this doesn't have to be word for word exactly what I have down here, but make sure you have the main valid points. So the remark, go and search for snails or whatever your organism is. I think another one, excuse me, is beetles. But I've come across students mentioning beetles, but I would teach snails. So mark each one with an inconspicuous mark on the back, or a mark on the back. So we're looking for your mark in them on the shell. So it's important that the mark does not harm the snail or make it more visible to the predators. Okay, so make a reference to that. Record. So record the total number found and marked. Place them back in the same position. So that's your day one. So day one, that's your capture. Sorry, your recapture. So a couple of days later. We're not going a week because what's the chances of those snails still being there? Very slim. So 2 to 3 days later. When you mention and reference your second visit, do say how long you went back. So I went back 2 days later, okay, on my second visit. So you're going to return to your same habitat, taking note of the number of organisms found and the total of these snails that were recaptured. It is very important that you say the word recaptured. Your first visit is your capturing. Your second visit is your recapturing. So therefore, you're making reference to your capture, recapture. Sorry. So release all snails back into that habitat, and then you're going to calculate out the population size. So make sure we say release, you're back into the habitat. So to calculate out the population size is capture on the first visit, and then capture, multiply that by the second visit, and of the second visit, how many had a mark on the back of their shell? Show them this, okay? You could even write this out if you want. Day one caught by day two caught. Sorry. Okay. Over how many were recaught. So you could write out that formula in your own words, whatever is easier for you. I'm just going to get a glass of water. Thank you so much. Sorry. Note to self, next time make sure I have a bottle of water here. I usually do actually when I record, but anyway. So here's an example. On day one, I caught 40 snails, marked and released. On day two, I caught 40 snails, of which 25 had a mark on their back. So just keep it really simple. You could say on day one, I caught 3 snails. On day two, I caught 3 and 1 had a shell mark on its back. Just keep it very, very simple. So where are marks going for? Marks are going for naming your animal. Marks are going for mentioning capture, recapture. Your first visit, you mark the animals, you record, and you place them back. Second visit, or 2 to 3 days later, that is implying that you've gone back again. Total number found, and you could say some of these had marks on their back, so these were recaptured. Reference has to go to recaptured. So release them back, calculate out your population. So marks will be going for calculating out your population and say what your formula is. Okay, they do like asking this. Now just be aware as well, the quantitative survey of animals or plants could be asked as part of the long question in section C, which would be very nice. It's popular that way as well. Okay, so now your abiotic factors. Abiotic factors hasn't been asked for a while. I did say to my students who set the leaving cert in the summer, be aware that abiotic factors has not been asked for a number of years. It could come up. So I'm saying, I'm not making a prediction. I'm just saying, be aware abiotic factors hasn't been asked for a number of years. It could be in section A, it could be in section B, or it could be in section C, but it hasn't been asked. So we need to know 3 abiotic factors. I'm going to have. Okay, so we need to know 3 abiotic factors. I have 4 here. So if you're mentioning temperature, say it's ground temperature or say it's air temperature. Don't say temperature on its own. So ground temperature, what do you use? There will be a mark going for a thermometer. So there will be a mark going for your apparatus. So you would place this in the soil, leave for 5 minutes, come back and record. So make sure you mentioned there that you're recording what the temperature is. Repeat this 2 more times in 2 other areas. What you need to be aware of. So what's the influence of this temperature? So temperature can affect metabolic rate of organisms. Higher temperature in the summer, more plant growth. Lower temperature, high animal activity. Lower temperature in the winter, and animals may hibernate or death, may lead to death. So with your abiotic factor, what influence does that have within your habitat? Okay, so that's important to be aware that they may ask. Now, air temperature is the same thing. So air temperature, I hung a thermometer from a tree. I left it there for 5 minutes, came back, recorded the temperature, repeated this in 3 other areas within my habitat. How does temperature affect what grows there? So the same thing. Your soil pH. So what we do in school is I would use a soil pH probe and I would collect a sample and do this back with the class. So soil pH, if you're doing the soil pH with the probe, we're going to put it in an area in the soil, leave it for 5 minutes, come back, record the pH. Repeat this in 2 other areas. If you did universal indicator, if you collected it and actually filtered it back in class, make sure that you mention that you got soil from 3 different areas. Now, so I have here the pH probe and then the pH meter. So if you gathered soil from your area, you would have mixed it with a bit of water, filtered it, and then with your filtrate, you would have added a universal indicator and checked to see what the acidity is. So for me in night school, most of it's between 7 and 8. So what's the influence of soil pH within your habitat? So favour some plants and therefore some animals, really. So that could be your 3. And then you have light intensity. So here I have an example of a light meter. So open it up, expose it to the light, record it there for 5 minutes. Open it up to the light, leave it there for 5 minutes and record. And I would encourage my students when they're doing light intensity to go to 3 areas. One that's in the light, one that's in the shade, and one that's exposed to a bit of both. So how does light affect, what's the influence of light within your habitat? So plants are adapted to different light intensities. So remember, plants need light to photosynthesize. So abiotic factors, you need to know 3. Hasn't been asked for a while. What apparatus do you use to measure your light intensity? How do you measure it? And be aware of the influence that the abiotic factor has within your habitat. And if you're asked this, I haven't just mentioned that you do this 3 times. Okay, so you want to get the average. Is this notes in the book? And no, this is not. Some of the pictures are. So what I'm going to do is, I think we'll try and make these available later, or if not, I'll put questions 8, 9, and 10, so all the experiment stuff is together. But we'll make this available later today or someday, soon. Okay, so that's your light intensity. Now, with ecology again, they may ask you to construct a food chain. So this is just from the textbook here. And remember, if you're asked a food chain, it must be arrows from one organism to the next. They can also ask you to construct a food web. Okay, and they may ask you to list some food chains within your food web, or they may say what would happen if the woodlouse or this field mice, what would happen if this field mice died off? What would be the effect on the rest of the food chains? They could ask you to draw a pyramid of numbers, and don't forget about your inverted pyramid of numbers, which I should have put in here. And on your inverted, what it doesn't do is take into account the size of the organisms or the number of organisms. So that's your disadvantages with your pyramid of numbers. If it's your pyramid of numbers and you have something large at the top, make sure you know that this is going to be your parasite. So that's ecology. So reminding you, Ecology could be asked in questions section A, section B, or section C. Right, so then we're on to the microscope. So you have to be familiar with the microscope, your light microscope. So I'm assuming now, I have not put that part in, I'm assuming you are familiar with it. So for your animal cell, this is going to be your cheek cell that you're going to look at. So you're going to swab the inside of your cheek with a cotton bud. Okay, so there'll be marks going for swabbing the inside of your cheek. You'll smear this across the glass. Okay, so there'll be marks there for saying this is put on your glass slide. Excuse me. Now, there'll be marks definitely for what stain did you use, or else they'll ask you, oh goodness, or else they may ask you, what is the stain that you use to test? What is the stain that you use when looking at an animal cell? They may also ask you, why do you use a stain? What's the purpose of the stain? Why do you need the stain? So the stain will make the nucleus appear darker. Okay, that's your answer there if they ask that. So then you apply your mounted needle at a 45-degree angle. Why? To prevent air bubbles. So this could be split up into two questions. How do you apply a cover slip at a 45-degree angle? Why do you apply it this way? To prevent air bubbles. Or it might be as why or how do you apply a cover slip at a 45-degree angle to prevent air bubbles? Again, read the question carefully and see if that's two separate questions. Here, sorry. So then you add your cover slip. They might ask you what is the purpose of your cover slip, to protect the specimen, to protect the lens as well. Now, that's your preparing. Now, how do you examine? So you're going to place your slide on your stage, secure it with the clips, and then you're going to always look with the lower magnification first. So yeah, there's that typo again. So low magnification first. Coarse focus, rough image, fine focus, precise image. And repeat this then at a higher magnification. So you can see here if this is at a low magnification here, and this would be at your higher magnification. If they asked you to draw it, you're drawing a circle there with just 3 labels, only 3. Okay, so that could be like back to Kevin's question. Draw a diagram of what you see. So that would be your cell there with your nucleus, your cytoplasm, your cell membrane. Yeah, that's your 3 things. And that it's circular in shape for your animal cell. Then your plant cell. So for our plant cell, our source is an onion. So you would pin off as small as possible layer, peel off your layer of your onion, your epidermis. I would get my students to, anything that they peel off to put into a Petri dish, excuse me, anything that they peel off to put into a Petri dish of water. Why water? To prevent the specimen from drying out. And I would tell them to try and get a few pieces, because see which one is the best. I would then advise my students to transfer that specimen with the paintbrush. So it's much easier to transfer with the paintbrush, and you can literally paint it onto the slide and smooth it out, rather than a tweezers. I know some of you might use a tweezers. I think a paintbrush. Now, if you said using as tweezers, place this onto the glass slide, it's still accepted. But I think a paintbrush is easier. So you place your specimen onto the center of your glass slide, and we add iodine. So this is important. They may ask you, what's the stain that you use to look at plant cells under light microscope? Why do you use this stain? So the nucleus will appear darker, and for iodine, it makes the cells more visible. How do you apply your cover slip? So using a mounted needle, apply at a 45-degree angle. Why? To prevent air bubbles. What's the purpose of a cover slip? To protect the specimen, protects the lens. Then it can do. Will the midterm workshop be streamed online? Yes, one of them on Wednesday. So I have 2 classes on Wednesday. I don't know which class, but it will be streamed online. So Wednesday we'll be making it. I don't, if it's live, I don't know at the moment. I'm not too sure, but I'll find out, and then we can let you guys know if that will be live. But it's definitely going to be recorded on Wednesday. Which class? I don't know yet. Okay, examine. So the examining is the same as the animal cell. Place your slide on your stage, secure it with the clips. Lower magnification first, lower magnification first, coarse focus for rough image, fine focus for precise image. Repeat this at a higher magnification. If they ask you to draw what you see, you're labeling your cytoplasm, cell membrane, cell wall, nucleus, and you can put in your vacuole. Do not write mitochondria, okay? Or chloroplast. They are only seen with the electron microscope. They're the ultrastructure of the cell. Please make sure your diagram is rectangular in shape. Your animal cell is circular. These are the only organelles here. So so the first thing again with the diagram, we look to see, does it look like what we're asking you to draw? And then we go with the labels. If you labeled chloroplast, it was a couple of years ago, the chloroplast, I think we just gave them 0 automatically. Sorry, if the student said chloroplast, it was 0 automatically. Now I think we just take a mark away from the label. So if there was 3 marks going here for the, sorry, it'd be 1, yeah, 3 ones. If 3 marks going here for the label and you wrote chloroplast, the max you could get would be 2. But an onion is a bulb, it grows in the ground. Onion is yellow in colour, it's not green, so therefore it has no chloroplast. It cannot photosynthesize. And then to isolate DNA from plant tissue. While this was asked, it was asked in the summer, and we all had a bit of an inkling it was going to come up. What's the probability of coming up again? I would say very slim. Just don't ignore it. Make sure you are still familiar with the steps. So to isolate DNA from a plant. So you could use a kiwi or you could use an onion. In school we use an onion. We don't use kiwis because actually a lot of students are allergic to kiwis. So we don't use a kiwi. So I'm going to go, this year they were particular about giving the students the marks for this. So there was marks going for chop, cut up your kiwi or your onion. Okay, so there's marks going for that, a reference to that this year. There was marks going, we wanted table salt and washing up liquid mixed together. Again, you do not need to put in a measurement in biology of how much table salt, how much pH, whatever it is. You don't need to say what the measurement is. So add the kiwi. So into a beaker, you have washing up liquid and you have salt. Add up your chopped kiwi, your chopped onion. So we're chopping it up because we want to open up the surface area. Add the kiwi to the beaker and pour in your salty washing up liquid. Put the beaker in a water bath at 60 degrees for exactly 15 minutes. So this summer, you had to have mentioned 60 degrees for 15 minutes. If a student said 60 degrees on their own, they got nothing. So they wanted a reference there to time. So when we do look at experiments in 9 and 10, we'll go through this bit here, but they were so at the beginning of the conference, at the beginning with marking, there was marks for table salt on its own, there was marks for washing up liquid on its own, there was marks for 60 degrees on its own and 15 minutes. And then they were like, no, we're actually changing this. So then they said, we want table salt and washing up liquid referenced together to get the mark. We want 60 degrees and they must say 15 minutes referenced together to get the mark. I've got another question from Ali. Will all the midterm tasks cover the same topic? Oh, so what I'm going to do for the midterm is genetics, and I'll talk about that just at the end of the moment, but it'll be the same in each class for that, but I'll come back to that, Ollie, and thanks for asking a question. I appreciate that. Okay, so 60 degrees in the water bath, and then we allow it to cool. So after cooling it, we're going to blend it for no more than 3 seconds. So they had to reference in this summer, blending it for no more than 3 seconds. And then they would have filtered it using coffee filter paper. And we use coffee filter paper because it's more porous. So then with your filtrate, you put that into a boiling tube or your test tube. And we add 2 to 3 drops of protease. And very slowly down the side, you're going to add ice cold ethanol at an angle down the side of the boiling tube. Now, they were very, again, this summer, if a student mentioned ice cold ethanol, they did not get the mark. They had to have referenced that it was poured slowly or trickled down the side of the test tube at an angle. So they're like, you can see now how they are very strict on some things. So the student had to have mentioned ice cold ethanol trickled down the side of the test tube. That was their 2 marks. If they said ice cold ethanol added to the test tube, 0. So they got, no marks for that. So then what will happen then, your DNA should become visible at the interface. So DNA appears as white, oops. I do not want. Okay, so DNA appears a white visible string. And then how do you extract your DNA? You can use a glass rod, or some of you use an inoculating loop to remove, there we go, to remove that. So glass rod or an inoculating loop. With the steps, they may ask you, why do you use salt? So salt is going to clump the DNA together. Washing up liquid. Why do you use washing up liquid? So washing up liquid helps to break down the cell membranes. Why do you chop it up into small pieces? You chop it up first to break down the cell wall. When we break down the cell wall, that's going to release the cytoplasm, and that's going to increase the surface area. Because remember, we want to get in to find the DNA. So think about it. We have our cell wall, our cell membrane, our cytoplasm, your nucleus. Your nucleus is surrounded by the nuclear pores. We want to get into it, so that DNA. Why 60 degrees? And that's to denature the enzymes DNases to get the DNA out. Why 15 minutes? If we do any longer than 15 minutes, you could permanently break down that DNA by the temperature. In fact, it could be, by the temperature. Why do we allow it to cool? We allow it to cool as it slows down the breakdown of DNA. Because remember, we want to get the DNA. Why do we blend it? We want to break down that cell wall if it wasn't fully broken down before to release that DNA. Why do we use coffee filter paper? It's more porous, so we should be able to get the DNA. So the lab filter paper pores are too small. Coffee filter paper is more porous. Why do we add protease? Protease breaks down the protein and the DNA. So remember, chromosomes are made of DNA and protein. Why do we add ice cold ethanol? So that's to take the DNA out of the solution. So our DNA will be visible at the interface. And it's ice cold ethanol, it's not ethanol. So your ice cold ethanol that you would be handed by your teacher should have come from the freezer. So it's ice cold ethanol at an angle, slowly down the side of your test tube. So that's your questions for question 8, all the experiments for question 8. So bear in mind that some of these questions can appear in section A, section B, obviously section B, and ecology could appear in section C, and DNA could appear in section C with genetics. Maybe not this, because we were really strict on it in the summer. So I'm just going to go back to Ollie's question there with the midterm classes. So in the midterm classes, what I'm going to cover is genetics. So that's chapter 12 for those who have the book. So you all know genetics is long. The class is 2 hours, and I will not get everything covered. But my plan is to look at DNA quickly, DNA and RNA, DNA profiling, DNA replication, protein synthesis, and then genetic crosses. And I think I have genetic screening. What I won't get done is evolution and variation, and I don't think genetic engineering. So if you have your book, it's chapter 12. If you don't, that's fine. So what we have is you will get a handout of most of that chapter. What you won't be included in the handout, because I won't get it covered, is evolution, variation, and genetic engineering. So the plan is that we will go through that. I'll have a PowerPoint to go through stuff. You'll be able to underline and write, and then together, when near the last part of the class, will be crosses, and we'll do genetic crosses together. Some of you may have done genetics, some of you may not have done it yet, but hopefully when the time comes that you do, if you haven't done it yet, when you do it in class, you'll have some understanding for it. What we would do in school is actually, we hold genetics off now until the end, will be the last chapter that we would cover. And we would give students an option of saying, look, this is what we do. I'm not speaking for other schools. We give students the option I'm saying if you'd like to do genetics, okay, you're to listen. If you don't want to do genetics, you're studying in class time. But genetics, I love it. One of my favorites. It's actually probably one of my favorite topics. I love teaching this. Love reading about it. This is the way. Look, this is the way the science is going. Like some of you are going to be studying this field, but yeah, I love genetics. That's why I put it that there. So look, I thank you for listening. First live one. I'm hoping now that our Monday grinds after midterm will be live, and like this you can ask questions, and always ask questions. No question is too silly. So I look forward to seeing some of you, I hope, on Wednesday or Thursday. And any other questions? I'm just going to give it a minute. Was it good? Did you learn anything new? Any feedback? Any questions? Oh, you're welcome, Alexandra. Thank you. Guys, I wouldn't ignore abiotic. I have a bit of my gut says abiotic could come up. I wouldn't ignore abiotic factors this year, is what I'm saying. Okay, so listen, thank you very much, and I will hopefully look forward in person to seeing some of you on Wednesday and Thursday. So thank you. Goodbye.